Yeah, this is yes, you've how long have we got? I'm gonna summarize about 40 years of interest and work. So I have been fascinated about how light is detected and how it regulates rhythmic systems. In fact, my PhD work was actually on birds and how they detect day length, which then, the increasing spring day lengths trigger reproduction. So there's a wonderful example of how light is being used to regulate a rhythmic process. And I was fascinated about the photoreceptors, the light sensors that birds use for that. Um, if we move to the mammals and the work we did on mice, it always struck me as being curious that the only organ that is used for the detection of light in mammals to regulate the clock is in the eye. And we know that because if you cover the eyes of a mouse or a human, or if you have no eyes, then the clock will continue to tick, but it'll drift through time. So most of us, for example, have a body clock which is slightly longer than 24 hours. And so, under constant conditions with no eyes, for example, you would get up a little bit later and later and later and later each day. And we've worked extensively with people who have lost their eyes. And what it emphasizes is the fact is that the eye has a dual function. It's the organ of space. It gives us our sense of vision, but it's also the organ of time because it's that light that is picked up by the eye that regulates the clock. And when you talk to individuals who've sustained massive trauma to the eyes, they sort of say, "Look, I can cope with not being able to see, um, but what I can't cope with is my body constantly trying to fool me." And I was told one really rather poignant story of this chap had lost his eyes, but he was determined to work out ways in which he could overcome this loss. And so he was determined not to have the messy front garden. So he'd worked out a way, he was completely blind, to go to the shed, get out the lawnmower, to cut the lawn, and there were little things he could push up, and so he knew he'd come to the edge of the lawn, turn it round. And so he'd worked all this out, just amazing. And on one occasion, his wife came down and tapped him on the shoulder and said, "It's 3 a.m. in the morning. You're waking up the neighbors." And we kind of smile at that. But, when you're trying to gain control after this traumatic loss, you can do it in lots of ways if you're blind, but you can't if you've got no regulation of the clock. But the point I'm trying to make is that the eye is doing two things. It's regulating our vision, of course, but it's also regulating our time. And the question we asked really quite some years ago now is, how does it do that? The visual cells in the eye, the rods and the cones, give us our sense of space. And we wondered, well, presumably it's those photoreceptors that are regulating the clock. But for various theoretical reasons, I wondered if that was the case. Because what vision has to do, and the rods and cones, is grab light in a fraction of a second and then make an image of the world. It's taking a snapshot of the light environment in a fraction of a second. But what the circadian system needs is an overall impression of the amount of light in the environment over relatively long periods of time. So, anyway, we started looking at mice with hereditary retinal disorders. These mice were being explored to try and understand the genetic basis of blindness in humans, relating key genes to visual processing. And we looked at mice who had these gene defects and were visually blind. And we looked at the effect of a light dark cycle on their ability to regulate their clock. And what blew us away was that these responses, the circadian system, could still be regulated by the light dark cycle in the absence of any visual responses in these animals. And it wasn't that they had some residual capacity. These mice could do this with the same sensitivity as those mice that had rods and cones. And anyway, we did 10 years of study on this, and there was huge opposition. I remember at one meeting, standing up and saying, well, these data are consistent, with the fact that the eye could well contain another unrecognized light detecting system. And at this point, somebody stood up in the audience, looked at me, and I thought they were going to ask an early question. They just said bullshit and walked out. I mean, it was ferocious, in fact. I mean, I remember one person said to me, "Are you seriously expecting us to believe this? We've been studying the eye for 150 years, and you're telling us we've missed an entire light sensor." Of course, I was relatively young in those days, full of confidence. I said, "Yes, based upon the data, that's it." And of course, from that point on, ultimately it led to the discovery of this third light sensing system within the eye. So the ganglion cells of the retina, they send their axons out of the eye, they form the optic nerve, and that goes into the brain. And what we discovered in mice and other researchers in monkeys and rats, and we also discovered this in humans, that a small number of those ganglion cells is directly light sensitive. They are photosensitive. So we've got photosensitive retinal ganglion cells, maybe 1%. And what they do is form this light sensitive net encompassing the arc of the eye, and they're used to detect environmental brightness. And it's that information that goes into the master clock in the brain, alters the gene expression of that molecular clockwork, aligning it to the outside light dark cycle. So that's what I've been thinking about for some considerable time, along with many colleagues. And I have to say, it's been an exciting time because the people that scream bullshit um, have now been involved very graciously, giving me some wonderful awards for the discovery of a completely new light sensing system within the eye.